Hi everyone, my name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia in Rockville, Maryland. I want to thank you all for joining me for today's webinar on improved promoter cloning technology for understanding gene regulation. We really appreciate it. Before I get started, I just want to take care of a few practical matters. First, you may ask questions at any time. But you will not be able to speak vocally to me. Instead, in order to ask a question, please type the question in the questions box in your control panel. If you cannot see your control panel, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of the buttons will be orange with a white arrow pointing in one direction. When you click on that button, you'll then see the control panel. Secondly, in the interests of keeping the flow of the talk going smoothly, I will wait until the end of the talk to answer your questions, in which case I will answer them in the order in which they were received. And finally, I will probably send you a link or two during the talk. I'll tell you that I'm doing that, of course, and I'm going to do it by pasting it into the chat box and then sending it to you. And then you can click on those links to open up your web browser whenever you feel like it. All right, so with that, let me get started on the talk. So what are the goals of this presentation? First, I'm going to give you an overview of promoter reporter technology. And secondly, I'm going to tell you about GeneCopoeia's vast collection of reporter clones with high-performance reporter technology. All right, so I'd like to start off with a brief survey. It's a one-question survey. The question is, why are you attending this webinar? Please take the time to answer quickly by typing the answer in your questions box. A, I want to use promoter clones and want to learn more. B, I heard about this technology and I'm just curious. C, I plan to use promoter clones and want to see what your company has to offer. Or D, other, and please explain. Okay, so let me tell you a little bit about who we are at GeneCopoeia and what we're about. We started about 15 years ago based on a large collection. Actually, the collections were intended to cover the entire mouse and human transcriptomes of ORF-cDNA clones. And what these are, they're ready to express human or mouse ORFs and a few from other species in mammalian systems. And as a result, we've generated a vast database for generating these ORF clones. Since then, though, we've branched out into other product lines, most notably promoter clones, which of course you're about to hear about today. A number of solutions for RNAi, like shRNA clones, qPCR reagents and arrays, genome editing tools like CRISPR and TALEN, and then some services associated with these stable cell lines, proteins, and lentivirus. However, most of our services still are connected in one way or another with this large ORF clone collection. Of course, you're here today to hear about promoter clones, so let's talk about that. All right, so here's the outline of the talk. First, I'm going to tell you about promoter clones and some of the technologies. These would be the standard or traditional reporters, and then the newer dual reporters. Then I'm going to give you some examples for applications for promoter clones and how GeneCopoeia can provide you with the promoter reporter clone that you need. All right, so why are promoter clones important? Well, one reason is in the wake of the post-genomics era, that is when the first drafts of the human genome and then later, of course, the mouse genomes were released, it became clear that we next needed to do functional studies, either looking at expression of the genes at the RNA level or function of the ultimate gene product, the protein. And of course, not to trivialize this, but the sequence, determining the sequence was the easy part. The functional studies and transcription studies are much more challenging. So in light of this, one way in which we've addressed this is to come up with a large catalog, of course, of human and mouse orphs, and by extension, human and mouse promoters. So for transcriptional regulation, these are great. So for the human promoters, we have a collection in our database of more than 20,000 promoters, and we have more than 18,000 mouse promoters. All right, so how do we define a promoter? Normally, a promoter has been functionally defined through experimentation, and this are usually elements that are proximal to the transcription start site, or TSS. These are TSS proximal DNA sequence elements required for native gene expression. Now, I'm not including the TATA box or any general transcription factor binding sites. I'm talking about elements that, the promoters will include the TATA box, of course, but they should also include elements for, say, tissue-specific expression of that particular gene, or of, or maybe, sequence motifs for hormone binding or such. Because of the proximal nature, this often excludes any distal enhancers. So you might be getting the native promoter, but there may be other faraway sequences that can influence gene expression. However, most mammalian promoters have not been functionally defined. So for our promoter clone collection, we instead take a more unbiased approach by generally using a sequence that encompasses approximately a little over 1 kb upstream of the TSS, then to about 200 base pairs downstream of the TSS. And these links do vary a little bit, but this is the general scheme. Okay? All right, so why would you use promoter clones? What are the general applications? These are just a few examples, but you might want to study regulatory patterns of gene expression in different tissues. Another is you might want to determine regulatory proteins that bind to a promoter sequence. Third, you might want to determine binding elements for activator and repressor molecules, like I said, a hormone or some other chemical ligand. Or another interesting application uses biosensors for physiological or environmental stimuli. Now, traditionally, the earliest reporter promoter clones took such a promoter sequence. Either it was already functionally defined, or it was one of these cases like I just mentioned where we arbitrarily determine a sequence, and just that sequence is hooked up to a reporter like GFP, a fluorescent reporter, or firefly luciferase, for example. So the putative promoter region of interest is cloned upstream of the reporter ORF in an expression vector. And then expression is followed either qualitatively, that is by, if it's GFP, for example, you look for green cells, or quantitatively, just by measuring the actual activity level, the readout level. Well, at GeneCopoeia we have such clones for traditional reporter technology. This is an example of one of our vectors where we clone a promoter, like I said, upstream of GFP, and these can be transfected very conveniently into mammalian cells. Here's a case study of published in a paper using one such traditional reporter clones from GeneCopoeia, and they used they studied two promoters, FAB-P4 and NRF2, which were hooked up to cyan fluorescent protein and YFP protein, respectively, to assay the responsiveness to alpha-mangosteen in adipocytes. Now, alpha-mangosteen is actually it's a protein from mangoes, and so this group was studying to see in what way this mango protein influenced gene expression. And so for the Fab4, shown in this is data from the paper. For the Fab4 promoter, there was no effect at all of alpha-mangosteen expression. And this is a quantitative assay. However, they got a clear result showing that alpha-mangosteen downregulates the NRF2 promoter. And so this was another clear example of how a promoter-reporter clone coming from GeneCopoeia can give a nice clear result in promoter regulation. All right, however, there's one significant limitation of traditional reporters like these, and that is quantitative measurement is actually fairly difficult. One must lyse the cells, and you cannot measure in real time. So if you wanted to assay different time points or different concentration points, you would have to come up with a whole bunch of, you would have to make a whole bunch of samples that you would then lyse at each point. However, we at GeneCopoeia have another product line known as the GLUC-ONTM dual secreted reporter promoter clones. And these have a number of distinct advantages. First of all, the promoter reporter readout is Gaussia luciferase. And Gaussia luciferase is great because, well, one reason it's great is because it is secreted into the medium. Okay, so you do not need to lyse the cells like you do for other reporters, including firefly or vanilla luciferase. So you can actually use the same cell, use the same sample multiple times to get a readout. You just isolate the medium. Also Gaussia luciferase is 1,000-fold more sensitive than both firefly or vanilla luciferase. And this system in particular actually has a second reporter, secreted alkaline phosphatase, driven by the CMV promoter. And this is a constitutively expressed secreted reporter used as a normalization control. In addition, this plasmid system is compatible with our own brand secrete pair dual luminescence assay kit. So this is basically how it works. So the promoter, which again is in our database, is arbitrarily defined with a little over 1 kb upstream and a couple hundred bases downstream of the transcriptional start site, is hooked up, is cloned into this plasmid to drive GLUC, Gaussia luciferase, and then it's simply transfected into cells. You add your stimulus, like I said, some kind of ligand, and then you measure just in the medium both the secreted GLUC or the secreted alkaline phosphatase. And again, in conjunction with the secrete pair kit, this gives you a very convenient promoter reporter assay. So these the vectors are transfected into cells, and then anywhere from 48 to 72 hours after the experimental treatment, that is the addition of your stimulus, you collect the culture medium and then set up assays in a 96-well plate and measure with a luminometer. It's very convenient. And this is in-house data that we generated using a number of different promoters, well-characterized promoters like EF1 alpha, GAPDH, etc., in this system in 2 different cell lines. So we found that we got good responses for all of these, and they also gave very different levels of response in different cell types, which is really not that surprising. So this is a nice result. So here's another case study, and again, this is using our GLUC-ON dual promoter clone. And in this case, in this paper, they were analyzing stimulation of the PIR-B gene by FXR, which is the bile acid receptor. So what happens is they activate the FXR using a synthetic molecule called GW4064. That activates, again, the FXR gene, and then they're looking for downstream activation of the PIR8 promoter by the FXR protein. And when they did this, they found, again, if they treat the cells with GW4064, they found a moderate stimulation 48 hours after transfection. But then 72 hours later, they saw a much larger response. And again, this is an indirect response to 4064, showing that the FXR protein can stimulate this promoter. All right, so now I want to break out a little bit from the PowerPoint and tell you how you can order GLUC-ONTM Promoter reporter clones from GeneCopoeia. All right, so I'm going to exit the PowerPoint briefly. Okay, and I'm going to open my web browser. And this is our home page. So what is done here is that if you want to search, we have a very convenient search function, and it's broken up by product lines. So if you click on promoter, you then are brought up with an advanced search page. So for most needs, you only need to use this top search function. So let's say I select human and I type in SOX2, press search. Okay, now what you see on screen is an accession number for SOX2 and a product ID. So when you click on the product ID, you then are presented with a number of different options. And these options range from the type of reporter you want, like you can get Gaussia luciferase by itself, or GFP like I mentioned, or you can get the dual secreted kit combination plasmid, which is the one we of course recommend. It's the scientifically highest quality product. And so, and also with these clones come in both non-viral and lentiviral format. So then, just for illustrative purposes, I'm going to select for purchase the non-viral and the viral dual promoter clones. Okay, so I've selected those, add them to the cart. Okay, and you need to create an account on our website first, so I'm going to log in. Okay, and I also, this is what comes up, and so the list price, these are the prices for the actual promoter clones. The non-viral format is $565, and the viral format is $695. And almost all of the promoter clone prices are the same. I also have some control, GAP-DH promoter control, as well as our secrete pair dual luminescence assay kit, which you can also buy here, and that's currently on sale for $303. So then all you have to do is check out. So anyway, that's the simple way of ordering promoter clones from our website. There are also some other services available that you're presented with, and also I should mention that if you don't want to use one of our arbitrarily derived promoter regions, you can always tell us what sequence that you want to use as your promoter, and that would involve getting a custom quote. You wouldn't be able to order on the website, but it's completely doable. All right, so let's go back to the PowerPoint. Okay. All right, so I'm also, this is a list of a select list of publications using our promoter reporter clones. Most of them use the dual assay kit. These are just 12 of them. So it's nice to see that your products being used in publications, of course. And then, okay, and so now I'm just going to briefly summarize. So I told you about how promoter reporter clones are useful for many applications in the analysis of gene expression. I told you about how traditional promoter clones use fluorescent reporters or firefly or vanilla luciferase, which work well, but they require cell lysis for quantitative measurement. I also told you about the GeneCopoeia brand GLUC-ONTM Promoter reporter clones, which offer higher sensitivity and ease of use over traditional fluorescent and luminescent reporters. And I showed you how customers can easily purchase GeneCopoeia GLUC-ONTM reporter promoter clones by searching our website database of nearly 40,000 human and mouse promoters. All right, so now don't just listen to me tell you. Please visit our website to get a first-hand look. This is our GLUC-ONTM Promoter Reporter clone website, and I'm actually going to send you the link for that in the chat box right now, so you can feel free to visit it if you want. I'm going to paste this in, and I just sent you the link, so look for it in your chat box. All right, so I'm just about at the end of the talk. I also want to tell you about another upcoming webinar. We give webinars on various product lines often. This is not on the Promoter Clone Web Talk, but there's a lot of interest in this on CRISPR and TALEN genome editing product lines that we have. And this one's entitled CRISPR and TALEN in Mammalian Cells, What Do I Do Next? So it's 2 weeks from today, Wednesday, September 24th. This is the registration link, but of course I'm not going to expect you to memorize that link. I'm going to send it to you in the chat box like I just did. And then all you have to do is click on that registration link and then register for that webinar just as you did for this one. Okay, so that's it. I will just I will stop here and take questions. I hope you found this webinar informative and useful. And if you have any questions, just go ahead and type them in the chat box, in the questions box. Sorry. Okay, so we have a question here. Do you have promoter clones for other species? If you mean species other than humans or mouse? We don't have a database of those. However, you can certainly order them if you give us the gene information, and we can make them in the very same fashion that we do for our mammalian promoter clones. Okay, are there any more questions? If you have them, go ahead and type them now in your questions box. All right, if not, then again, I want to thank you very much for attending the webinar. If you have any additional questions, you can call me. This is the number, 1-866-360-9531. My extension is 227. And again, my name is Ed Davis. Or you can email me, of course, at edavis@genecopoeia.com. I will send you my email address in the chat box. So that you should be able to just click on, and it'll open your email. Okay, and something came through. And that's it. So again, I want to thank you all for attending, and have a great day.